And now it is my great pleasure to introduce Dr. Paulin Jirkof.
Thank you for the invitation and for the introduction. And we wanted to start with a short survey before I start with my talk. After my talk, you will understand maybe why I want to ask you these questions. There are 4 questions, and at least those that work with animals, I would ask to answer these questions. If you don't understand it, if you've never heard of the concept that is asked for, then just say no. Okay. So I leave you a little bit of time to answer those questions. They are all somehow related to refinement and about things that we will talk about. And I will then, at the very end of this talk, we will share the results and discuss them. The topic of the talk today is how small, and you can replace that also with simple. Refinements can improve the life or the lifetime welfare of laboratory rodents. So it will be rodent focused, but many of the concepts I will talk about are actually also very useful for other species. Just have to adapt them a little bit, species typical. And I'm not going into detail on what I mean with lifetime welfare, right? Very shortly, I want to talk about refinements in every part of the life of the lab animals, therefore breeding, handling, housing, and so on. So, and I think welfare is something that is something that is not a short-term experience. It's something that is for sure a long-term experience. This is why I choose these way to describe it. But we will not talk that much about lifetime welfare because this is, of course, a concept that we would have to talk about. So, first of all, my definition of refinement. Refinement is everything that avoids or minimizes actual potential pain, distress, or other adverse effects experienced at any time of the life of an animal. On the one hand, so reducing suffering. But on the other hand, it's also about enhancing animal welfare, enhancing animal well-being. This is important because if we accept that these animals do feel pain, so we accept they are capable of feeling emotions, then it is not just about negative emotions. Of course, it's also about positive emotions. Just think about your own life. Just being without pain, just being without severe distress does not mean you're happy, right? It's the balance between positive and negative experiences that count at the end. Most of the things I will talk today are real refinements. That means they are above that is asked for in the legislation, no matter if it's German legislation or Swiss legislation. And I will talk about going beyond that, taking the extra mile, because that is, in my opinion, welfare or refinement in the sense of welfare. Many of the things I will talk about today, some of you will know about, right? So some of you will know about all these methods and tools that we're talking about, and some of you will use all of them. And if that's the case, you can be proud of you. It's a very nice thing. Many of these things and many of the publications I will show you today are 10, 15 years old. And one could say, well, there is no use in talking about this again and again. Nevertheless, also my experience from the last 10, 15 years when it comes to refinement in laboratory animal housing, handling, and experiments is that most of these refinements are not used routinely. At least they are not reported in the literature. And because literature reporting is always a thing, right? It's not clear if something is not written in a publication, and it's not clear that it's really did not happen during experiments. So you cannot always be sure and cannot rely completely on these kind of systematic reviews, for example. This is why I'm always interested also in doing some surveys, some formal, some more informal like today. So we will talk about your use of simple refinement measures later. But I will show you also some results of systematic reviews. So this is what I'm going to do today, or these are the topics I want to talk today about. For sure, that is the refinement of experimental procedures, but very, very important also the improvement of routine handling and routine housing things, procedures. I will also talk a little bit about meaningful monitoring of strain or suffering or severity, and about applying mitigating measures to refine or to reduce this strain. And we will do a little bit of a reality check later. So I will show you some of the results from our systematic reviews on the actual use of refinement. Good.
So what I'm going to do is to have some examples, typical examples for simple and non-expensive and easy refinement approaches. And one thing that everybody who works with male or with mice may have encountered is the problem of housing male mice. So mice are social animals, the females and the males, no difference. But we all know if you work with mice, male mice do have some or show internal male aggression at a certain age. And this is an issue because this can lead to stress in the animals, and it can lead to severe injuries. Here, for example, that is, I would say, a more severe fighting injury that you see here. What you normally would do now is you would single house these animals, you would separate these animals. Single housing, of course, is detrimental for social animals, and in this case for the males. They also have social needs. They also have some thermal needs. So thermal regulation is actually hampered when they are housed single. So there are good reasons to try to avoid single housing. Sometimes the animals have to be removed from a study or have to be killed, which of course brings other problems for science and for animal welfare. But I would argue that we actually should separate them in many cases much earlier than we normally do. Because when you see something like this, what happened before is normally this. So what you see here is the pelt of an animal. This is a CD1 male, and it has been pelted to the backside of the fur. What you see here are bleedings, hemorrhages that come from fighting. All these strange red dots here are actually fighting wounds. I don't have a picture of the other side, but I saw all of these animals. So you can believe me, on the other side, there's perfectly white fur of the CD1 mouse. So we tend to separate them actually not early enough. There's very likely already a lot of fighting going on when we normally separate them. But separating them, as I said, is not the solution because of the negative effect that they might have on animal welfare. And so if you have this, separation should only be the ultimate ratio, right? So you should try everything to prevent the aggression in the first place. And especially for male mouse housing, there are there's a whole list of things you can do. We can try. Might not work in every case, but it is something that is worthwhile trying. One example would be to use less aggressive strains because there's a genetic component to this. Use stressful procedures. We know that after stressful procedures, male mouse fighting increases. Using tunnel handling and predictable handling, is something that we in Zurich have a lot of good experience with. So tunnel handling can reduce male mouse aggression. What we also have very good experience with is this transfer of nest material, used nest material to the new cage. These are things that are described nicely in the literature. Same is true for age allocation, the use of siblings, having stable groups. There is also a lot of discussion about group size. We once did a study with American colleagues. We found for CD1 mice, or CD1 males at least, that groups of 3 had less fighting wounds than pairs would have. But there's also a crowdsourced study from NC3Rs which actually claims the opposite. So what I want to say with this, there are a lot of things you could try before it comes to aggression, and you should try these on a routine level to avoid aggression or to delay aggression as much as possible. Another example when it comes to housing. We all know environmental enrichment is important, and cognitive enrichment is important for all animals that we house, giving them the opportunity to experience novelty and so on is important to enable natural behaviors and to promote positive affective states. But if we are honest, the standard housing systems, standard caging systems that we normally have in the facilities offer really limited space for this. So they are really restricted. They are not ideal for housing mice and rats, if we are honest. And they reduce the possibilities we have for environmental enrichment. If you cannot change the way your animals are housed, you might install something like playpens. I know this is not something that is routinely used yet for rodents, but we have very good experience with that for cats and for dogs, actually. And there are even publications on playpen use and the positive effects of playpen use in mice. We in Zurich do have some groups that offer temporary playpens for rat groups, for female rats in this, or female and male rats. And this works nicely. It's always difficult to assess the real animal welfare effect on it. As I said, here in this study, they analyzed it. They have an increased and approach latency and more anticipatory behavior, which is all a good sign. They also observe more ambulatory behaviors than non-ambulatory behaviors in the playpen animals compared to the control animals. So all of this can be a hint that this really is an advantage for the animals. And if the hygiene allows it, this is a very nice and easy way to offer more cognitive environmental enrichment for those animals. Housing is something that all animals experience, and handling is something that probably also most animals somehow experience. And most of our lab animals are actually prey animals, so small prey animals. Mice, for example, are animals that suffer in the wild from predation from birds or from larger mammals like foxes, for example, and therefore picking them up from above and by the tail is coercive. There are studies showing this. This again has been published in 2010, right? Also together with the solution for this problem, the use of gentle handling methods like cup or tunnel handling. It makes sense to use tunnel handling methods because mice and rats too are tunnel digging and tunnel living or dwelling species. So it's very natural for them to enter tunnels. They feel very safe in a tunnel, and you can use it nicely for transporting those animals. We have, in my lab, I don't have a lab anymore, but in the last years when I still had a research lab, we only use tunnel handling, and I supported many groups in the transition from tail to tunnel handling. And I can tell you, if it's done right, it really is a nice thing. I often hear it takes longer to use tunnels, but this then normally is a problem of technique. So if you know how to do it and you do it right, it's actually as fast as using the tail. There are studies showing that there are positive effects of this, like, for example, this one showing that tunnel-handled and cup-handled animals do interact more voluntary and longer with human handlers, so they get more tame. There are also some studies who wanted to show that and positive welfare effect. That is not so clear. The problem is not that this might not have a positive effect, but the problem is that we are very bad in assessing positive welfare. So we might have to work on the methods we have to assess positive welfare until we really could assess how good that is for the animals. Nevertheless, it's for sure not harming. And if you ever tried it, it really works well. Talking about handling, let's talk also about experimental procedures. So for most animals, as I said, handling, the simple restraint already is something that is aversive. This old data showing an increase in heart rate after a few seconds of restraint. There are other studies who could show that there is an increase of corticosterone when you perform simple standard techniques with these animals that somehow are based on restraint. And especially if you then add something on top, for example, for drug administration, that could be something that involves some stress for the animals. Yes, mice can get used to something like this. also rats and other animals. But especially these very small animals like mice might just accept it, but they still will not like it. So thinking about ways to, for example, reduce the frequency of injections, so the need for restraint, or maybe even thinking about alternatives to replace those techniques that might be stressing for the animal, can be a very simple way to reduce the stress in the animal. So think about all the alternatives to reduce the stress of administration procedures when you plan your experiment. And I will give you some examples. First one is, of course, thinking about reducing frequency of administration. This is an example from pain treatment, pain relief, from older paper from our lab. This is about buprenorphine, typical opioid analgesia that is often used in small animal medicine. And what you see here basically is the target concentration after the injection of this opioid. You experience a fast increase of the serum concentration under the target concentration that you actually aim for, which means you have to re-inject every few hours. So picking an animal up, injecting that is already in pain, that is something that can induce pain, even more pain, and definitely some stress. So reducing this frequency would be very valuable. And there are some alternatives, technological solutions actually. And one are so-called sustained release or depot formulation. So you actually load a matrix with a drug. In this case, in this example, again, it's buprenorphine, and then you have a continuous release of the drug into the blood into the organism via this matrix for several days, 24, 48, 72, depending on the formulation you use. This means you have a continuous, secured, stress-reduced administration because you do not have to administer several times a day, but just over 2 or 3 days. Unfortunately, sustained release formulations are, until yet, mainly available in the US, so there you can buy them and use them. This is the reason why in Zurich, University of Zurich and Basel together developed a sustained release formulation for rodents, which actually had a quite good characteristics profile in the analgesic test here, better than the normal one, but even better where the measured serum and brain concentrations, so we could really show a prolonged release of these drugs. Here in this drug, in this case, brain concentration of the drug with the depot formulation, with the standard formulation. Don't misunderstand this data. Yes, this is the target concentration for the serum. And you could, of course, argue, well, the normal formulation also has a quite high level in the brain. And nevertheless, what we also found out is that this serum concentration obviously is a much better predictor of pain relief than the brain concentration, or that we need higher concentrations in the brain for buprenorphine to have an effect. So in Switzerland and in the US, you can get these formulations. That would be a pity for you in Germany. I don't know if something changed in the meantime, but we had now the very good progress in Switzerland that we can probably very soon also import the American drugs into Switzerland for use in Switzerland. If this import is possible in Switzerland, I'm relatively sure this is also possible in another European country. So if you're interested, you should try to get such an import license, because this really is a very nice refinement. Alternative, if you cannot get these kind of technology that solves your problem, is to reduce the stress that is linked to the administration. And what I show you here is a very, very nice video. This is from a Swedish organization. This is a subcutaneous injection in a mouse without a restraint. And if you know a little bit about mouse stress signs and pain signs, you see that this mouse is relatively relaxed. This is the result of many days of training. So probably nothing that you would do in routine in most experiments, but it should be a very good example on how you could how much you can achieve with good habituation and training. And if you can do that in these little mammals, it's probably also very easy to do in larger species if you work with a larger species. If you're interested how these kind of habituation processes work, there are very nice videos of another Swedish organization called RISE for mice and for rats. I'm happy to send you the link afterwards. I don't have it now here, if you don't know it. And the third option, of course, would be to replace stressful procedures completely. And one example is the replacement of oral gavage. And this is an example from a colleague of mine, this is a rat that is trained to ingest a tasty mixture of a drug. So it's a drug mixed with something that these animals like as an alternative to an oral gavage. For those who are not sure what an oral gavage is, you use a tube that you put down the esophagus of an animal to get a drug directly into the stomach. This, you don't need a lot of creativity and empathy, or fantasy and empathy, let me say like this, to know that this is for sure stressful. And here in this video, you see a very nice example for a refinement procedure. The rat in this case is actually enjoying what happens. You see it at the very end of the video, it doesn't want to stop. So no stress involved for the animal, and additionally, a little bit of reward. And also very interesting, of course, or very nice that the person actually handling this animal does not have to know a lot about certain procedures, right? Just feeding an animal is relatively easy. So you don't need a person that got training to get competence in a certain invasive technique. This is basically animal handling. So a win-win for everybody involved, actually. It does not only work with rats and larger species, of course, it also works very nicely with mice. This is the so-called micropipette guided drug administration. What here is used is basically condensed milk, which is then mixed with the drug that the animal should ingest, and it's given with a pipette micropipette, which means you have complete control over the intake of the drug in the animal, and the animals learn that very, very fast because they really like condensed milk. It's sweet milk. Here, another example how that could reduce stress. So this is plasma corticosterone level in native animals compared to those that receive restraint and IP injection. You see a significant increase in the stress hormones, while during the MDA procedure, there's obviously no release of stress hormones. This is something for sure that's very intuitive when you see also how the animal is, how relaxed the animal is ingesting the drug. So these are very simple methods. You can also use other matrices or carriers. Nutella is something that is described. Baby food works very nicely in rats. Strawberry jam very nicely in mice. Honey. But if you are afraid of all these substances and that they might interfere with your experiments, what also works very nicely are sterile calorie-free jellies that you could use. Keep in mind, this might change the bioavailability of your drugs. So you have, if you change from IP injection to voluntary oral ingestion, there might be chemical or pharmacological interactions, there might be hygiene problems, but all of these normally can be solved with a little bit of creativity. Be very careful with uncontrolled protocols. What I showed you now were controlled protocols, but for example, medicated drinking water is something that you cannot control, and mice do have a typical circadian rhythm with these are drinking events of several mice here, individual mice with less intake during the day and more intake during the night phase. So if you want to have a continuous intake or an intake at a certain time point, then uncontrolled intake via drinking water is for sure not something to be recommended. When we talk about refinement in housing and handling, we should also talk about how to measure refinement effects. So what are there? What positive effects do I see? What negative effects do I see of my procedure maybe? Or I want to assess the impact of the procedure in order to apply a refinement, right? And for this, I have also prepared a few slides because I think it's super important to talk also about meaningful assessment of strain. And assessing effective states in rodents is not that easy. One of the reasons is that we very often use very unspecific parameters to assess something like this. This is an example for body weight changes. Probably everyone uses that, and it is a very useful parameter, sure. Not in all cases. This here are animals, the first three groups that do not receive any painful procedure. These last three all received surgery, and this group here actually did not receive a painkiller. But you do see there's actually no reduction in body weight because this is a minor surgery and you do not see the pain effect very often in the body weight development. So this is why it is super important to always use specific parameters, and especially for pain, we do have specific parameters. This here is a composite score, same groups of animals, composite score designed out of different pain-specific indicators that then very nicely actually detect the problem. And on the other hand, you can, of course, choose indicators that are more sensitive to positive affective states, but this is unfortunately still quite rare. But let us talk now about the negative things. If I talk about specific indicators in our composite score, for example, for pain, because we work with abdominal surgery, we use specific abdominal pain signs. These are some prominent examples: stretch, press, writhing. You will see all these behaviors in this video. If you have another surgery, you might choose different parameters. Again, pain-specific parameters for that different condition. Here, after an osteotomy surgery, limping, guarding does not have to be, that can be a pain sign that you could actually detect with this more specific sign. Whatever you do, try to find parameters that are actually evidence-based and scientifically validated. That is not so easy. Again, here for my example, abdominal pain. And we used a lot of burrowing activity because burrowing is a natural behavior of mice that is reduced when these animals have abdominal pain. We could show that in phlebotomy. We could show that in DSS enterocolitis. Other groups could show that in mouse cholestasis. But there are studies showing that in other abdominal pain conditions like mouse colitis associated colorectal cancer, it is not a good sign or a good tool. So make sure you find your meaningful indicators for assessing any type of welfare reduction or welfare impact. Then, last step, applying mitigating measures. So what to do to make it better for the animal if you see that there is reduced welfare. And here we really have to talk about score sheets. That could be a good tool to do that. That should then trigger predefined actions like an increased monitoring frequency, analgesia administration, or fluid therapy, like I see it here. And they also should trigger for sure humane endpoint. Not going into detail here for the sake of time. I think you know what I mean with this. A typical measure that is supportive of mitigating is analgesia. Careful, of course, analgesia is not a refinement, right? Analgesia is a legal must. You have to use analgesia if you have a painful procedure, but you can have good and you can have bad analgesia. So use tailor-made analgesia protocols and ideally use multimodal approaches. This is the gold standard for large animal medicine. It's the gold standard for human medicine. It has many advantages because there are synergistic effects. You use, for example, a systemic pain relief and additionally a local anesthetic. Then you have a maximization of the pain relieving effect. And in many cases, you have minimization of the side effects because you will need less analgesics when it comes to volume dose. Yeah, that is basically a very fast forward summary of some important refinement measures that are all relatively easy and cheap to get. I promised a little bit of a reality check, and I can do that because in the last year we spent, me and other colleagues of mine spent a lot of time trying to assess the actual use of refinement measures and of monitoring measures in systematic reviews. And I give you now some examples. So it's a lot of data, so I will only give you a few examples that are dealing with things that we have talked about in the talk. So first of all, are methods to assess affective states, positive and negatives, actually used in the lab animal field? We did a review, a narrative review on this in several systematic reviews. I can already tell you, well, yes, there is an increase of use of these methods, mainly concentrating on the negative affective states like pain, which makes sense because that's much easier than assessing positive affective states. The problem is very often the absence of negative signs is interpreted as being positive, right? That's for sure not really true. Very likely the absence of negative states or signs is probably indicative of normal function. There are several methods that I used more and more that are promising. Activity, vocalization, but also facial expressions. So when we talk about mice and rats, for example, the mouse Grumman scale or the rat Grumman scale, and this is data from a systematic review where I could be part of the team. And you see, this is just for surgical interventions in mice and rats. And the question was, are mouse Grumman scores used more often in mice or in rats since they have been developed in 2010 and 2011? And the answer is yes, but take a look at the numbers here. I mean, this is a very specific question, so post-surgical pain, but the 1, 2, maybe 8 publications a year is not a lot. So there is potential, even so we can be optimistic. Reporting of refinement as such is not so nice, so not so good. Let me say that this actually it's quite grim. If you would say, if you take a closer look, then let me say like this. This is from a not yet published study where we analyzed the reporting of refinement in DSS-induced colitis models in 2 different time spans. Fortunately, in none of the publications we took a look at was the term refinement used in the sense of the 3F. There was in none of the publications assessment of well-being or pain described. There was a little bit of health monitoring, a standardized one, but nothing that would be indicative for well-being or pain. And we had very rare reporting on humane endpoints or analgesia. When I mean rare, I mean we had 2 publications that mentioned analgesia actually, and DSS-induced colitis could be painful, right? We can argue about that if it's really painful. Maybe depends a little bit on the protocol. But what is for sure painful is ECG telemetry transponder implantation. This is a major transplantation surgery. Here we checked all publications from until 2019. Again, very rare reporting of refinements, very rare reporting of welfare and pain assessment or humane endpoints. Rare in this case means, for example, humane endpoints were reported in 3 publications. Analgesia, and they were not even called that, right? It was more a description of when animals were killed. And then we had very incomplete reporting of analgesia. Very rarely multimodal analgesia was used, even so this would be a very good model to use multimodal analgesia. even more shocking, we even had some publications that mentioned that animals do not need pain relief after this kinds of surgery, and therefore they did not receive it. Good. As I said it before, systematic reviews only help us to show what is reported. So I'm always interested also in doing some surveys and being part of it. The first results you see here was a survey that we did with our National Swiss 3R Competence Center on non-invasive genital handling methods. We had 231 responses, and we asked the people if they were aware of non-invasive handling methods like cup and tunnel handling, and if they use them. And there are still some people who have never heard about this, even though this is 10 years after the first description of tunnel handling in a scientific paper. Most of them are fortunately aware of these methods, but only you really use them regularly. Another survey that was an international survey from the UK with a little bit more participants came to very comparable results. There are still a lot of people who use tail handling only, or tail and non-reversive methods, and very few that only use non-reversive methods. Good. We will take a look at your data later to sum up, and so that we still have some time for discussion left. If I want to phrase it positively, we do have a great potential for improving the overall welfare in and outside of animal experiments. There's a lot of potential to still apply refinement measures. If you want to do better, or if you want to do the best, what I would suggest is to have an action plan on refinement for your specific experiments or for your specific facility. First of all, inform yourself about advances in the field, especially when it comes to assessing effective states and when it comes to analgesia. There's a lot of progress in the last years, very interesting field at the moment, a lot happening there. Do train yourself and train your collaborators, including your animal caretakers, including the PhD students and so on. Challenge always the status quo. Just because something is happening since years, it's not good, and it cannot be. And all these things can be changed. Don't be too much of a perfectionist. So perfect is the enemy of better. So if you can do a little bit better, that is good. That is important for the animals. It doesn't have to be perfect. It doesn't have to be completely, I don't know, animals. There might still be residual pain. There might still be a little bit of stress involved. That is better than having a lot of stress and a lot of pain, right? Find creative ways to solve the problems with all people involved. Test your options systematically and have alternatives in place whenever something might not work. If tunnel handling is nothing that can be implemented in your facility, think about cup handling. And if you are successful with something, share your experience, talk about it, and also publish it, right? Publish it in your materials and method sections at least, because otherwise new scientists or scientists who will try something new will not be guided in a meaningful way. People have to read about refinement to think about them, to know that this is a standard maybe in the field, or make it a standard in the field. Good. That was actually my talk. I will stop now. I hope the audio was now much better than before, and we still have the survey. And I would actually now maybe stop the survey and share it with you, and then we can open the discussion. So just for summary, many here use gentle handling techniques. Very good. 20 to 30% not. That is regular in those groups that are actually interested. And there are even people using slow release. So you definitely have to tell me which kind of preparations you use. And again, many using monoanalgesia, and also quite a large group using multimodal analgesia, which is nice. I hope that we see these every year that the numbers are increasing. And then 50-50 for the grammar scales, and very few we use methods to affect just as positive affective states, which something we could maybe discuss, because I would really be interested in working with those who use it, what type of methods they use. Maybe you can just write that into the question. Okay. So now, how do we do it now? We'll just go to the question.
Yeah, there are several questions already, and you can see them in the Q&A section. So maybe we start with those.
So should I just well, there's one question by Reiche. What is the red mouse? What if the red mouse do not like to voluntarily eat the solution, or it's not a gavage, right? It's voluntary. What is plan B if you cannot mix it with condensed milk? Do the animals take it? Plan B, yeah. So plan B is, first of all, find something that masks the taste of whatever you have to give the animals. Right? There are some things that are nasty. Tamoxifen is one example. Tamoxifen is obviously not very tasty. And I do have mixed reports of people that use it with condensed milk and it works, and for others it doesn't work. So you might have to try with the dosage, and you have to try with the different carriers. If it doesn't work at the end, you will have to do a classical oral gavage, right? I mean, it is an animal experiment. At the end, it is important that you get your experiment done, and you don't want to waste animals for a not successful experiment. So that would be my answer to this. I hope that this answers a little bit. Are there any?
Maybe one question to add on to that. So how, because you mentioned it should mask it, how do you know that? Do you try them yourself, or I mean?
That is best practice, actually. So if I give analgesics in the drinking water normally, I do try that, because I want to know if it's bitter, if it's sweet, if it's strange. But of course, you will see it if they love condensed milk, they love soy milk and all this stuff. And if they don't ingest that, even after a little bit of training, when your substance is mixed, then it's probably very nasty stuff. So I wouldn't taste tamoxifen. I didn't want to try tamoxifen and things like this. But for analgesic, I would do it, actually. Yeah. Then there is another question here on, do you try to do subcutaneous injection without fixation with CD1, only with CD1 mice, or also with other mice strains that are not as calm? I didn't do it at all, to be honest. This is something that is from this Swedish group. They work with all different types of strains, but of course there are more calm strains that are easier to handle, and CD1 are super nice. But also Black 6 mice, they are a little bit more reactive, but they are also very nice. So if you have a good habituation of the animals, normally it's fine. There are some jumpy strains, some very stressed strains that you might not be able to train. Then again, for the safety of the experiment, you probably will decide to choose the classical method. So, what drugs would you recommend for DSS experiments? I think it's about analgesic drugs, which do not interfere with inflammation. Reducing inflammation would mask the phenotype. Exactly. And many analgesics do somehow interfere with inflammation and the immune system and other things. So whenever you have, this is true for most models, that you have to check if your drugs do interfere with your model. There is actually a study on the side effects of different drugs, anti-inflammatory drugs, antipyretics, and opioids on DSS-induced colitis. It's not yet published, but I know it's in review. So I can tell you already there are drugs that do not interfere with the classical DSS colitis phenotype or effect, and this will be published very soon. Good. Can you please provide a list of the mentioned studies? This is Alf Galuske. You mean the mentioned studies on the systematic reviews? I'm not sure. I can definitely do that. Can also share my slides, by the way. So if that's something, can definitely share the PDFs. Then is a German question here. Lauten, so should I translate that in English? Die Umwidmungskaskade für Tierarzneimittel in Deutschland. Okay, this is a comment saying it will probably be possible to get this kind of sustained release formulation imported into Germany. It wasn't possible in the past because the sustained release formulations were not did not have a complete FDA approval. So they had this very specific solution that they have for experimental use in animal experimentation in the US, but this has changed now. It has a full FDA approval. So yes, I am sure that this is possible now to import. I'm not sure. I assume it might be possible to import into Germany. So try it, because in my experience, these sustained release formulations are super helpful for everyone involved, right? Good thing. And then, okay, the question on this regarding the citations of my slides. So as I said, I'm super happy to share the PDFs with the full citations if that is of interest of someone.
So thank you again very much, Ms. Jirkof. I also have one question. You mentioned that it's much harder, of course, and that's less frequently done to measure positive welfare and effects. So how would you actually validate this? Because, I mean, we measure stress hormones and all other things for negative effects, but how can you measure positive ones?
So for other species, so larger species, there are some very good examples. Not all of them are, I mean, everything is context specific, but play, vocalizations, some facial expressions have been described in many species to be very good to assess positive effect. Same is true for some activity measures. For mice and rats, it's a little bit more difficult. But play and ultrasonic vocalization work also in rats very nicely, right? We have ultrasonic vocalization that is actually linked to positive affective states. Play is also something that happens in rats. It also happens in mice, but not under standard conditions, at least not in my experience. I only know it from pet mice. And then there are, of course, these super sophisticated tools like cognitive bias tests and things like this that can measure mood in animals. But it is difficult in mice and rats. I think at the moment people are working mainly on vocalization and also positive facial changes in mice and rats. And this can be combined with artificial intelligence and machine learning approaches. I think that's very likely the future of assessing affective states in this smaller level.
There's one more.
Oxytocin as a marker for positive well-being is the question. What's my opinion on this? In the past, I was super excited about oxytocin measurements, I have to be honest, because, of course, oxytocin is somehow related to positive social bonding and all these things, and has measurable effects on well-being in humans, likely. But I never worked with it, and I have not seen that much convincing research on it. Maybe I missed that. I'm not sure, because I never worked with it. But theoretically, I think it's a super nice idea to use oxytocin for measurements of positive affective state, but I'm not aware of any good examples that have been used for applied refinement research, to be honest. So, not sure.
Okay, if there are no further questions, we will finish the session and thank Ms. Jirkof again very much for joining us today. And so have a nice evening and goodbye, everyone.
Yes, thank you very much.